Hello students, I am teacher Svetlana and in our today's lecture we will learn about the platelets and blood clotting and about the structure of our heart, which is covered in the chapter Respiration and Circulation. So let's begin. Students, thrombocytes are cellular fragments formed from the large cells called as megakaryocytes. They are produced in the bone marrow. They are very small oval-shaped cells, fragments without nucleus. Now have a look at this image here. This is your bone marrow, which is the site for the production of your RBCs, platelets and your white blood cells. This is how your different components of the blood appear. These are your RBCs, these are your WBCs, and these tiny ones are the platelets. The normal count of thrombocytes in human blood is about 2.5 to 4.5 lakh per cubic millimeters of the blood. Students, now if the number of the thrombocytes decreases than the normal condition, it is called as thrombocytopenia. This condition causes internal bleeding, hemorrhage. Then you have the platelet secrete platelet factors, which are essential in blood clotting. They also seal the ruptured blood vessels by formation of a platelet plug, which is also called as thrombus. They secrete serotonin and a local vasoconstrictor. So let us see the phenomenon of blood clotting. Clotting or coagulation is a process of converting the liquid blood into a solid form. This process may be initiated by contact of blood with any foreign surface, that could be intrinsic, or with a damaged tissue, which is an extrinsic process. Intrinsic and extrinsic processes involve interaction of various substances called as clotting factors by a stepwise or a cascade mechanism. There are in all 12 clotting factors numbered as 1 to 8. Factor 6 is not in active use. Interactions of these factors in a cascade manner, that is in a stepwise manner, leads to the formation of the enzyme thrombin. The thromboplastin helps in the formation of the enzyme prothrombinase. This enzyme inactivates heparin. Now, heparin is an anticoagulant which prevents the clotting of blood. So, they inactivate this enzyme and they also convert the inactive prothrombin into active thrombin. Thrombin converts soluble blood protein fibrinogen into insoluble fibrins. These fibrins form a mesh in which platelets and other RBCs are trapped to form a clot. Have a look at this image here. Here you can see that there is a rupture in the blood vessel and the RBCs, platelets, they are moving out from the broken vessel wall. Now what happens is when it is ruptured, then you have this entire event taking place where then you have the activated platelets which form these fibrin. This fibrin forms a mesh here in which the platelets and the RBCs get trapped and they form a clot. That is, they plug the ruptured region. This phenomenon is called as blood clotting. Here students, you can see how with the help of a flow chart the steps of the process of blood clotting is explained. You have the intrinsic pathway here, and we have the extrinsic pathway that's present here. In the extrinsic pathway, it begins with the damage to the tissue outside the vessel. Intrinsic pathway begins from the damage to the blood vessels. In both the cases, you have the inactivated factors are converted into active factors, and then you have two important components present in your blood, that is prothrombin, which gets converted into thrombin, and fibrinogen, which gets converted into fibrin. This thrombin inactivates heparin, which is an anticoagulant, and this fibrin ultimately forms the blood clot by forming a mesh and trapping the activated platelets and the blood cells. Now students, let us learn about the heart, which is the main organ of our circulatory system. Heart is the main pumping organ of the circulatory system. It is red-brown in color, hollow, muscular organ, roughly to the size of one's fist. that is when you close your fist, that is the approximate size of your heart. If the average weight of it is about 300 grams in male and 250 grams in females. It is conical in shape and it lies in the mediasternum. Mediasternum, students, is the space between your two lungs. It is broader at the upper end, and that is the base, and it is conical at the lower end, that is the apex. Conical end is slightly tilted to the left side and it rests above the diaphragm. So this heart is enclosed in a membranous sac called as pericardium. Here we have a diagram of the heart wall and the pericardium. This region is your pericardium, this is your heart wall. The pericardium is formed of two main layers, outer fibrous and inner serous pericardium. Serous pericardium is soft, moist and elastic. Have a look at these diagrams. Here you can see your serous pericardium. It is formed of squamous epithelium and it is further divided into two layers, that is your parenchymal layer and visceral layer. This is the parenchymal layer and this is the visceral layer of your serous pericardium. Parental and visceral layers of the serous pericardium are separated by a pericardial space. The space in between is called as the pericardial space. It is filled with pericardial fluid, about 50 ml, which acts as shock absorbers and protects the heart from mechanical injuries. It also keeps the heart moist and acts as a lubricant. Now students, inner to your pericardium is your heart wall. It is mesodermal in origin. Its wall is formed of three layers: outer epicardium, middle myocardium and inner endocardium. This here is the outermost layer, that is your epicardium. This in between is your mesocardium, the middle layer, and the innermost layer here is your endocardium. Epicardium is thin and formed of single layers of flat squamous epithelium resting on the basement membrane. Myocardium in the middle is a thick layer formed of cardiac muscles, and the endocardium again is a single thin layer formed of squamous epithelium. The epicardium and the endocardium are protective in function, whereas the myocardium is responsible for the contraction and relaxation of the heart. So these two, the outermost and the innermost, are having protective function, whereas the middle thick layer of your mesocardium is responsible for the contraction and relaxation of your heart. Now students, let us try and understand the external structure of the human heart. Students, the human heart is four-chambered. There are two superior chambers that are called as auricles and two inferior chambers that are called as ventricles. Externally, the atria, these are separated from the ventricles in this region by a transverse groove called as coronary sinus. Here, this is the coronary sinus, or it is also called as the arterioventricular groove. The two ventricles, this region, they both are separated from each other with the help of the anterior and the posterior intraventricular sutures. So here also the ventricles are separated. The coronary arteries and the coronary veins runs through these sutures. So here you have the coronary arteries and veins running. The pulmonary trunk arises from the right ventricle. So this is your right ventricle. From here the pulmonary trunk arises, and the aorta from the left ventricle are present. So this is the aorta which arises from the left ventricle. Now, the pulmonary trunk bifurcates into right and left pulmonary arteries. Aorta, which is also called as the systemic aorta, is divided into three regions as ascending aorta, systemic arch, and the descending aorta. The ligamentum arteriosum joins the pulmonary trunk and the aortic arch. So here you have this aortic arch being formed. Then it is a remnant of the embryonic duct called ductus arteriosus. Now this aortic arc gives out three arteries, which is your innominate artery, left common carotid and the left subclavian. The right atrium receives the superior and inferior vena cava. Here you can see this is your right atrium and it receives the superior and the inferior vena cava along its dorsal surface. Pulmonary vein opens into the left atrium along the dorsal surface of the heart. So here students, this is the left atrium and the pulmonary veins opens in this region. Now students, we'll have a look at the internal structure of the heart. Internally, we have the atria. They are the thin-walled receiving chambers. These two are your atrias. This is your right atrium. This is the left atrium. They are separated from each other by an interarticular septum. Interarticular septum has a oval depression which is called as the fossa ovulus here. It is a remnant of the embryonic aperture called as foramen ovulus. The superior vena cava and inferior vena cava and the coronary sinus, all these three, the superior, inferior and the coronary sinus, they all three open here into your right atrium. Opening of your inferior vena cava is guarded by your Houston wall, and the Tabascan wall guards the opening of the coronary sinus into the right atrium. 4 pulmonary veins open into the left atrium. These openings are without the wall. Have a look here students. This is your left atrium and you have the pulmonary veins which open up into the left atriums and they are not guarded by any walls. Now students, both these atriums open into their respective ventricles by the arterioventricular apertures. Now these openings are guarded by the tricuspid wall in the right atrium and the bicuspid wall in the left atrium. Now all these heart walls, they help in maintaining the unidirectional flow of the blood and they prevent the backflow of the blood. Ventricle students, these are the inferior thick-walled pumping chambers of the heart. The right and the left ventricles are separated by an intraventricular septum. Now students, the walls of your left ventricle is more muscular. It is about 3 times more thicker than the right ventricle. Have a look at this image here. This is your wall of the left ventricle, which is, you can see, quite thicker as compared to the wall of the right atrium. Inner surface of the ventricles shows several ridges which are called as column cartini or trabecular cartini, which divides the lumen of the ventricle into small pockets or fissures. Have a look at this. This is your trabecular cartini, which divides the inner walls of the ventricles into small pockets. The lumen of the ventricle also shows inelastic fibers known as chordae tendon. This here is your chordae tendon. These here attach to the tricuspid and the bicuspid walls, and they in the ventricular walls, they attach them to the ventricular walls. Here you can see these attachments. Yes, and they then regulate their opening and closing. Now students, the right ventricle opens into the pulmonary aorta and the left ventricle opens into the aorta. These openings are guarded by 3 semilunar walls each. These walls prevent the backflow of the blood into the ventricle. So this is the internal structure of our heart. Thank you students for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe and keep learning.